Chapter 7, Part 3.
Okay, ladies and gentlemen, welcome back. This is Chapter 7, Part 3, and I do believe I'm recording here. Aren't I recording? I think I am. Anyway, so when we were last discussing, the concept that the population death rate in any lethal treatment is constant, inasmuch as it is a percent per hour reduction, given that the treatment is A, lethal, and B, remains constant during the time that the bacteria are dying. So we saw that we can represent the death rate of a microbial population with an arithmetic or logarithmic graph, and that the concentration of the killing agent, the environment, the temperature, whatever, that we expose our microbes to might change the speed at which microbes die, but they will still die at a rate of percents per unit time, 90% per unit time, or any percent per unit time. Okay, such that a high population or low population load will increase the time at which it will take all the microbes to die, but they'll still die at the same rate given the same treatment. So, what kinds of treatments can do that? Well, as we talked about earlier, the actions of microbial agents, if whether it's heat or chemicals or whatever, there are some basic mechanisms by which you can get microbes to die. Membrane permeability, you change the permeability of the membrane. If you make the membrane very permeable, then everything will leak out of the cell and the cell will die. You change proteins, well then the cell can't do anything. Pretty much everything a cell does is dependent directly or indirectly on proteins. So if you damage those proteins, the cell won't be able to do anything and the cell will die. Likewise, you damage the DNA, then you can't make the protein. And if you can't make the proteins, the cell dies. So damage to the DNA, it can also be lethal. What kinds of treatments can cause these lethal results? Well, heat, radiation, and then we'll talk about a couple others. So these methods are physical rather than chemical, so heat. If we treat with heat, there are some terms that the thermal death point is the lowest temperature at which all cells in a culture would die within 10 minutes. Thermal death time, the time during which all cells in a culture are killed, how long it takes for all of them to die. And then decimal reduction time, the minutes to kill 90% of a population at a given temperature. So we start with 1,000,000, 90% die per minute, so in this case the decimal reduction time is 1 minute, because that's how long it takes to kill 90% of the population. After 6 minutes, we should theoretically at least have one survivor. How many survivors after 7 minutes? Everybody think about that question and then try to give an answer. How many? So this says after 0 minutes I have 1,000,000. That means when I start I have 1,000,000. After 1 minute I have 100,000, then 10,000, then 1,000, then 100, then 10, then 1. How many after 7 minutes? Everybody think about that. Alright, that's enough. Sorry. So, did you think about it? Hopefully the answer you didn't think of, or maybe you thought of this, but if the answer you chose was 0, you're not exactly right. Each time we have a tenth of the microbes, and yet if we wait 7 minutes, we're not guaranteed to have 0 microbes. The mathematical answer is a tenth of a microbe, but there's a problem with that too. If you have a tenth of a microbe, well, you don't have a tenth of a microbe. There ain't no such thing. Let's see.
Bedtime. This is not a dime. This is a dime. Break it in half. You don't get two nickels. You get shit.
You didn't hear that, did you? Anyway, so if you have a tenth of a microbe, you don't have a microbe. You have something other than a tenth of a microbe. And that was a scene from One Flew Over the Cuckoo's Nest. It's an important movie if you guys haven't seen it, and it does feature some expletive language. Nevertheless, so can you have a tenth of a microbe? No. Do you definitely have 1 microbe? No. Do you definitely have 0 microbes? No. You have, I guess the best answer is you have a 10% chance of having a microbe in there. So the idea of when you will be certain that all your microbes will be gone, you will never be 100%, 0.0000, etc. Certain that your treatment has given you sterility. It is possible to get sterility. It is not guaranteed that your treatment will give you sterility. And if you are selling cans of green beans that have 1 million Clostridium botulinum endospores in them per can, then the time that you treat that can before you sell it matters. So yeah, I think at this point I'm going to just move on. Okay, so which of the following best describes the pattern of microbial death? 1, the cells in the population die at a constant rate. 2, all the cells die at once. No, that's not the case. Not all the cells are killed. That's not always the case. It can be the case. It depends on the antimicrobial agent. Well, the speed can depend on the antimicrobial agent, but this answer A is true. Okay, so moving on. How do we get microbes to die? Well, let's look at some traditional methods, and here is the most common, the most traditional, and the most widespread method of just killing microbes, and this is the autoclave. If you're in a hospital, or you're in a microbiology lab, or you're at a community college, or pretty much anywhere, well, I guess certainly a hospital, your hospital depends on a autoclave, not a microwave, excuse me, autoclave to get sterility. There are some microbes that can survive autoclave treatment, but they are rare. Most of the time when you treat with a autoclave, you obtain sterility because those particular microbes that are able to survive aren't in your sample of whatever it is you're trying to autoclave. Surgical implements are autoclaved. Growth media is autoclaved. Other things, great many other things are autoclaved before they're used, and that is to obtain sterility. Autoclaves use steam, high temperature, and high pressure. You can see here there's a very thick door. This is basically a very, very large, powerful pressure cooker. There's a high pressure more than 1 atmospheric pressure, and then when you have high pressure, higher temperatures don't necessarily result in boiling. The boiling point of water goes down when you're at altitude and the air pressure is lower. It goes up when you are under higher than atmospheric pressure. So, in fact, you can go down to the bottom of the ocean and you can have very, very high temperatures, way, way above boiling, that where the water is still a liquid and not a gas. So here, autoclave is usually 121 degrees Celsius and 15 minutes, yeah? You can use dry heat sterilization. This will destroy microbes. But if there's no steam, there's no pressure, then just putting something into the oven, if you want that to achieve sterility, you're going to need to compensate with higher temperature and longer time than you get in an autoclave. So incineration, that also certainly, when you set things on fire, that is universally sterilizing. No microbe that I can think of survives open flame. Okay, so shall we put our, we want to make sure our foods are bacteria-free before we sell them. Can we put our eggs in here? Of course not, because if we did, we'd have hard-boiled eggs, and many people like to fry their eggs in the morning instead of hard-boil them. So, that brings up an important concept, which is that many, or reminds us of an important concept, which is that many treatments that obtain sterility ruin the things that we're trying to sterilize. We can't put eggs into an autoclave to sterilize them because that ruins the egg. Likewise, we can't put milk into an autoclave because the milk, while it doesn't turn from a liquid to a solid like the contents of the egg do, is still tangibly affected in its taste and to some degree in its nutritional properties when it's autoclaved. So, as a compromise between the need to kill microbes in our foodstuffs and the need to have edible foodstuffs that aren't destroyed by our sterilizing treatments, we have pasteurization. Pasteurization represents a sort of a compromise between obtaining complete sterility and destroying the food we want to sterilize. So, this is a heat treatment process which reduces spoilage organisms and pathogens and extends shelf life without the expectation of achieving sterilization. So, is sterilization possible with these treatments? Sure, if you have very few microbes in the first place, then you might find yourself lucky and get something to be sterile with pasteurization. But there's nobody expecting that. Pasteurization is done with the idea of reducing the kinds of microbes that spoil our milk and spoil our other foods. There's several ways to do it based on the kind of food you're trying to treat and other things, yeah? So, 63 degrees Celsius for 30 minutes, that's low temperature pasteurization. You can also do very high temperature, very short time, 72 degrees Celsius. You can get this equivalent effects in 15 seconds. And 140 degrees Celsius, this is called flash pasteurization. That can be done in less than a second, where many things die, but thermoduric organisms, very hard, tough organisms survive. What about vitamins? Well, it looks like if you look at things high temperature short time or ultra high temperature pasteurization, you get a little bit of loss of vitamin C. You get virtually no loss of folic acid, etc. It looks like there's a small amount of vitamin loss from these treatments, but not a significant amount of vitamin loss. Okay. Filtration. So, filtration is the idea that we remove microbes without harming solutions. Yeah. If we have growth media that we want to sterilize, well, perhaps the growth media has vitamins in it that, unlike these vitamins, will be destroyed by heat. So we can't autoclave it. Well, what can we do? Well, one thing we can try doing is filtering out any microbes, yeah? If you have a HEPA filter, right, and a HEPA filter is something that's pretty common. A HEPA filter removes microbes that are greater than 0.3 microns in size. Does that remove bacteria? If you remember the size of bacteria, you should know that they are generally about a micron in every dimension. And if that's the case, then if the pores of the HEPA filter are smaller, then the HEPA filter would remove bacteria. What about viruses? Well, most viruses are smaller than 0.3 microns, so viruses would be able to pass through, whereas bacteria and yeast would not. Membrane filtration, well, membrane filtration will remove significantly more viruses along with the bacteria. Still, there are viruses that can pass through a 0.22 micron filter. Other methods are only bacteriostatic. So, for example, if we put food in the fridge or the freezer, microbes don't die, they just stop growing. Yeah? So when microbes stop growing, then when you return them to warmer temperatures by taking them out of the fridge or out of the freezer, those microbes can wake up and start growing again. Yeah? So, when we apply low temperatures, microbes don't die. Those treatments are just bacteriostatic. Also bacteriostatic is desiccation and salting, right? If we dry or salt fish, we can cause plasmolysis of the bacterial cells, but then when water comes back to those cells, those microbes can wake back up. Okay, radiation. We'll talk about this in the next section.